we've learned that enzymes are super important in terms of how they facilitate different types of biochemical reactions that happen every day, every minute in our body. So let's take a look, though, at how actual enzyme regulation itself can take place. So first is just a definition. So we need to, regulation is kind of the key to almost everything in life. Anything that you talk about in terms of biology all comes back to this concept of how things get regulated. So we have to have certain things regulated throughout the day. We actually have kind of a rhythm of certain things being regulated throughout the day. So our enzymes, we've talked about this a little bit already, they themselves can be regulated by a number of things, such as some change in body temperature or a change in the pH in an environment. How certain molecules are going to be produced that might inhibit or promote the enzymes, their ability to work. And what's the role of things called coenzymes and cofactors. So all these things are things we've just mentioned, and now we're going to get into a little bit more detail. So when something inhibits an enzyme by specifically going into the area that it normally would do its job, in other words, its active site, and we call whatever that is something called a competitive inhibitor. So a competitive inhibitor has to have a shape that's very similar to the normal substrate of that enzyme. And so when it can go into the active site, it will slow down or inhibit the enzyme from doing a job. And we're going to talk about why sometimes those are important. There's also something called non-competitive inhibitor. And in this case, a non-competitive inhibitor actually goes somewhere other than the enzyme's active site. But in doing that, it's going to also change the rate of some reaction. And let's take a look at this again. So if it's a normal enzyme, we see that as the purple line here. This means that here in the beginning we have a low substrate concentration. So this could be like we don't have a lot of sugar, and then we're going to break down sugar, maybe one of the polysaccharides. So maybe at this point it's kind of low, and so our rate of reaction's low, our substrate concentration is low. All right, but now we're starting to get more and more substrate. So as substrate concentration increases, in other words, we're getting more of that polysaccharide, we're starting to see that the enzyme is getting busier and it's having a faster, it's able to have a quicker reaction. So the rate of reaction increases. Now why that's true is because when you have more of whatever it is that the enzyme works on, that it's substrate, there's more opportunity then for that enzyme to basically run into its substrate because it's like more crowded. So there's a point where that enzyme's going to be super active, and you get to the top of the curve here, what this indicates when it starts to flatten out is that whatever amount of enzyme is there is all being sort of used at that moment in time, and so we can't go any faster. So the reaction will continue, but it can't go any faster than that because there's not enough enzyme to go any faster. When we have something that's a competitive inhibitor, so it's out-competing or it's trying to compete with for that active site with that normal enzyme. So then what it does is the enzyme is still sometimes running into its normal substrate, but sometimes it's running into something that's competing, and therefore is in slowing down that enzyme's ability to do what it specifically is designed to do. So that's a competitive inhibitor. It may take longer for that enzyme to ultimately reach that point where things are breaking down at the same rate as the normal enzyme. So this is why this curve is lower. And then something that's a non-competitive inhibitor, which goes into a place other than the active site, is basically changing the shape of that enzyme a bit, and therefore when it's in that area, when it's changing the shape, that enzyme can't do a very good job either. So we even have a lower ability for that enzyme to do its job. And as you can see, it never reaches the maximum rate that that enzyme should be able to work at. And here's another little graphic I found online that I thought was kind of a interesting way of looking at it. So here's the active site again. Here's the substrate. Goes into that active site. That contact then allows the enzyme to do whatever it does, whether it's to strain the substrate or to cause some type of reaction to take place. Maybe it's hydrolysis. So we're going to break this thing now into these two products. A competitive inhibitor is basically, it gets into that active site. Sometimes it's there, and so it's going to slow down this enzyme. So once that happens, this thing will eventually leaves. It doesn't lock in there. It's not irreversible. It can reverse. A non-competitive inhibitor, you can see what's happening here. It's right down here, and it's kind of interfering with the activity of that enzyme in its active site. So one thing a new vocab that's a good one to know is this term allosteric. So allosteric is referring to a change of shape. So we have two types of allosteric inhibitors. One allosteric inhibitor is going to modify the active site of the enzyme so that the substrate binding is reduced or prevented. So that's kind of like what we just talked about before. So here's the active site. Here's the allosteric site, just meaning another place other than the active site. And this inhibitor, if it goes in and enters this allosteric site, has changed the shape now of the active site. Therefore, it can't pick up that substrate anymore. That would be an allosteric inhibitor because this is inhibiting the activity of that enzyme. However, there are some enzymes that actually need something to change their shape in order that they can work. And that's an allosteric activator. So here you see, here's the activator. Here's the non-active site. This is so it's the allosteric site. It goes in, and when it does that, it actually opens up this active site so that now this enzyme can do its job. So that's an allosteric activator. Now, they use a term down here, affinity. So it says modify the active site of the enzyme so that the affinity for the substrate increases. That just means that now it's able to do that. It has this ability now to pick up that substrate because it has been allosterically modified to change. Now, this is really important stuff, especially when we start looking at drugs, because when we look at medications, a lot of them are designed specifically to do something to the enzymes in a particular pathway. So some of them will inhibit an enzyme. Very often that's what they do. That's their job. They go in and we want to slow down an enzymatic pathway. We can use certain types of drugs to inhibit that pathway. That's one possibility. So we'll look at some of this. Another thing we mentioned is that some enzymes may not get changed allosterically by something, but they may need something to help them anyway. And these are called cofactors or coenzymes. If it's a cofactor, it's usually an ion. And that could include, in this case, DNA. If you remember, DNA polymerase was an enzyme that helped DNA to make another copy of itself, and it needs zinc. So that 2+ is what we're seeing here, zinc. Coenzymes are other things. These are things which we consider organic molecules themselves. Remember that these up here are inorganic, so organic things. These can include even ATP or some other things that we'll take a look at, and also include vitamins. So these can also be helpers. You might think of these as these enzymes, and these cofactors are helpers for that enzyme to do its job. Okay, great. So now we have this concept of enzymes. But now we're going to make it one more degree of complicated and to say that when we look at chemical reactions in biology, we're almost never seeing a very simple thing happening, one simple step. Very often what we see in many metabolic pathways is a whole series of steps that lead to ultimately some end product. So let's kind of follow this down here. When we have, here's the enzyme and here's the substrate, and now the enzyme's entering the substrate's active site. Something's happening here. The enzyme is doing some job. We get this kind of intermediate substrate that gets formed. Now we get a new enzyme, a new activity here, another intermediate substrate, another enzyme, another activity here, and then we get some end product that once this starts to build up can actually go back and inhibit the original enzyme. This is called a feedback inhibition pathway. And so in this case, what is happening here is that whatever this is now going to go back and it's going to temporarily inhibit this enzyme because whatever product was made was sufficient. We don't need more, so now we can regulate this enzyme by actually using some of the product itself to shut off the pathway. So this is an important concept to understand, that this idea that feedback inhibition is a process where the end product, once it builds up, once you get a lot of end product, some of that end product can serve as a way to inhibit or shut off the pathway. So this is a very important way of regulating. So in a lot of ways, if you look down here, what you see with all of these arrows is representing another chemical reaction, an intermediate chemical reaction to eventually get to this, which is our end product. So lots of different ways that we regulate in biochemistry. It's important, this is just to remind you that metabolic pathways are almost always some series of reactions with lots of enzymes involved. And then we have different ways to inhibit or activate these pathways. Lots of vocabulary too that was thrown in here. I started writing kind of a list of things like catabolic reactions, anabolic reactions. These had to do with the earlier videos, activation energy, enthalpy, entropy, and different terms for reaction. So we're going to build this vocabulary so that you feel as you're reading this that you're confident in your understanding of this information. So I think that's a pretty good stopping point. And we're going to look a little bit, I'll show you a video with you, I'll share a video of what happens, some diseases that involve problems with enzymes and enzymatic pathways. So metabolic disease. See you soon.